All right, great. Listen, welcome back. This is part 2 of our study of meiosis. In the previous segment, we looked at the different kinds of cells that we find in the reproductive organs, particularly of mammals. And of course, that includes you and me. And as we are picturing here, you can see we have these tiny little cells that have these long tails referred to as a flagella. And they're swimming around and trying to get into this larger cell that we see in the background, which is commonly referred to as an egg. Both of these, we said, are gametes, and they are genetically different and distinct from genetically speaking, they're distinct from the cells that we find in the rest of our body, which we refer to as somatic cells. These are haploid cells because they have half the genetic information. Now, in this segment, we're going to discover exactly how these are made to be genetically different from your body cells. And of course, this is all taking place within the reproductive organs. Okay? So first, again, some terminology that's going to help us make this distinction. There are really two sets of homologous chromosomes. Homologous chromosomes are two chromosomes, one inherited from the mother, one inherited from the father, that have the same length and general appearance. Again, these homologous chromosomes are kind of like your shoes. You know that they go together because of their general appearance and their size, but you can easily tell by trying to fit them on that they are not the same. You can't switch shoes, can you? All right? And we have copies of the same genes on those homologous chromosomes. Although these two copies may differ because they may carry different traits for each of those inherited characteristics. So they're not copies of each other. So homologous means similar, closely related, but it doesn't mean that they are identical. Okay? And you can see that especially with the sex chromosome. Both the X and the Y, here's the X chromosome and then the Y next to it, both of these chromosomes carry sex-related information, but they're obviously very different. And although we don't see the differences in the autosomes, the remaining 22 chromosomes, obviously they're going to be different in the same kinds of ways. Now, there's another set of chromosomes that we need to consider as well. And we refer to these as sister chromatids. And these are copies of the exact same chromosome. If you recall back in your days of studying mitosis, during the cell cycle, in part of interphase that we refer to as synthesis, copies of chromosomes are made. And this, of course, also occurs in our sex organs. So sister chromatids are duplicated chromosomes that remain attached to each other throughout most of this process of meiosis. And these originated from the same parent of the individual. You can see the distinctions set up in this illustration. All right, first, you can see pairs of homologous chromosomes. In other words, these are closely related chromosomes. They're similar in their general appearance, and yet they've both been copied during that process of synthesis, during the cell cycle. These chromosomes have duplicated because they're going to separate later on. Now, cells are going to go through 2 rounds of division in meiosis. We'll take a look at that. Meiosis reduces chromosome number and creates genetic diversity. Keep in mind that when we are studying mitosis, we started out with 46 chromosomes, didn't we? When we got into the synthesis phase of interphase, that number 46 jumped up to 92. We're going to see a similar process here. So we're going to be reducing the number of chromosomes to create the genetic diversity that we see in each of our families. And so in meiosis, like we said before, we have genetically unique cells. They are haploid cells, meaning they have half the genetic information that their original germ cells came from. And this takes place only at certain times in the life cycle of the organism. And these are the ones that are involved in sexual reproduction. So meiosis 1 and 2 are each 4 phases similar to that we find in mitosis. Now pay close attention here because there's some important differences. There are pairs of homologous chromosomes that separate during meiosis 1. And these homologous chromosomes, again, they're similar, but they're not identical. Now, the sister chromatids are going to divide from each other in the second round of 4 phases, referred to as meiosis 2. And these sister chromatids are copies of the same chromosome from the original parent, from each original parent. Again, this diagram may help you make those distinctions. So how does this work? Well, meiosis occurs after DNA has been replicated. And so you have, really, for humans, we have 92 chromosomes that we're speaking about. And they're now going to be separated in this process of meiosis 1. And what takes place is the division of homologous chromosomes in these 4 phases that you see pictured here. And the actual separation of these homologous chromosomes is going to take place during anaphase 1. So by the time we get to telophase 1 that you see pictured here, those homologous chromosomes that are closely related now have separated. And so now we go from a truly diploid state. In telophase 1, we're now in a haploid state because we have half the genetic information. In meiosis 2, this is when the sister chromatids, those identical copies that came from each respective parent, are now going to be divided. The DNA is not replicated between meiosis 1 and meiosis 2. Remember, we have plenty of chromosomes that have to be separated in what I showed you earlier. What's taking place here in meiosis 2, again, these phases are very similar to what happens in mitosis with these important distinctions. Again, it's in meiosis 2 that the sister chromatids are going to be pulled apart. In anaphase 2 here, that they're going to be pulled apart. And so what results at the end of meiosis 2 are 4 genetically unique gametes. And we'll see what happens to these later on in the next segment. Cytokinesis has taken place, and so these each have half the genetic information that will be found in this particular organism. And so they're not genetically identical, they're genetically very unique from each other. So meiosis differs from mitosis in some significant ways. Let's summarize. Meiosis has 2 cell divisions while mitosis only has 1. In mitosis, homologous chromosomes never pair up. In meiosis, this results in haploid cells. In mitosis, what we've looked at previously, that results in diploid cells. And side by side, you can see them. You can see the differences even more apparent here. Okay, so in mitosis, we're producing genetically identical cells. In meiosis, it's genetically unique cells. In mitosis, we're producing diploid cells that have the full complement of genetic information. In meiosis, it must be only half the genetic information because of what's going to take place next during fertilization. In mitosis, this type of cell division occurs throughout an organism's lifetime, even in an older person like me. In meiosis, however, this is only going to take place at certain times in an organism's life cycle. For example, you may have a pet, and that pet is only going to go into heat at certain times of the year. That's what we're referring to here. In mitosis, that's asexual reproduction. Meiosis, however, is sexual reproduction. So you can see these four differences as we place them side by side. Haploid cells develop into mature gametes. Let's study this further. We refer to this as gametogenesis. This is the production of gametes. And gametogenesis is going to differ somewhat between males and females. In sperm, they become more streamlined and they can move around. And you can see we refer to that as motile. Sperm primarily contribute DNA to an embryo. And you can see these pictured here. Now, in sperm production, we're starting out with a germline cell that you see here, which is diploid. We're going through meiosis, and so these are going to result in genetically unique sperm that you see here that are haploid. And as these mature, they're going to develop each the flagella that makes them highly motile. So from one germ cell, we're getting 4 sperm cells. Eggs contribute DNA, cytoplasm, and organelles to the embryo. So there's much more there that is going to be contributed by the female. And during meiosis, the egg gets most of the contents. The other cells are going to form polar bodies. Well, what does that look like? Let's take a further look. And so, like in the males, in the female, you can see that we're starting out with one germ cell that we see pictured here. And then beyond that, you're going to see a very different setup where only one of these potential gametes is going to have nearly all the cytoplasm, which is going to include mitochondria, stored energy, and so forth, into one egg. These polar bodies, however, are going to degenerate and they're not going to be useful for cellular reproduction. And so over the course of the lifetime of a female, they're going to produce far fewer viable eggs than the number of sperm that we see produced in males. Well, you can see there are some differences that occur in gametogenesis between the two genders. Thanks for watching.